Welcome to the deep dive. Okay, we're jumping straight into a pretty complex biological puzzle today.
Right.
How does a tiny organism, when it's really stressed like, facing starvation, how does it coordinate its defenses across totally different body parts?
Yeah, getting the whole system on the same page, like linking the brain to the gut.
Exactly. Not just a subtle hint, but a massive, system-wide, life-extending response.
It's like the ultimate survival package, really. We know about the cell's internal quality control, the unfolded protein response of the endoplasmic reticulum, or UPRER.
The ER being the cell's protein factory floor.
Precisely. And UPRER is the cleanup crew for when things get messy or stressed.
And the master switch for that cleanup is usually this factor, XBP1s.
Hmm?
Normally, gut stress activates it in the gut. Simple enough.
Mm-hmm.
But the source we're diving into today, this really fascinating paper on the little worm C. elegans, shows something else entirely. It's called cell non-autonomous signaling.
And that's the absolute core finding here. Activating XBP1s just in the worm's neurons. Well, that triggers the UPRER response way over in the intestine.
Wow, okay, so flipping a stress switch in the brain tells the gut, the body's metabolic center, to start cleaning house and boosting its defenses.
Exactly. And the payoff, it's huge. Better protein quality control or proteostasis, and remarkably, a dramatic increase in the animal's lifespan.
Okay, so our mission today, for you listening, is to follow that signal. We need to trace the path from that stress neuron all the way to the distant intestine.
Find that molecular messenger, the one acting as this global regulator of organismal responses to stress.
All right, let's unpack this. First step.
Yeah.
What's XBP1s actually doing in those neurons? It's clearly not just fixing local problems there.
No, it's definitely prepping them to send a signal. When XBP1s is active in a neuron, it switches on a very, distinct set of genes compared to other tissues.
Not the usual cleanup genes you'd see in the gut.
Not primarily, no. In the neurons, it's mostly changing genes involved in communication. Things like synapse function, GPCR signaling, how the nervous system develops. It's tweaking the wiring.
So XBP1s isn't shouting, clean up this neuron. It's shouting, rewire the communication lines. Prepare to send a system alert.
That's a great way to put it. It's remodeling the communications infrastructure.
Okay.
So once they saw that the gene expression profile was all about signaling, the hunt was on for the actual molecule being sent out.
Right. What's the message?
They screened a bunch of known signaling pathways, looking for that specific key needed to trigger the gut response and the lifespan extension.
And they hit on two essential molecules for this long-distance effect, acetylcholine, AD, and tyramine.
Yeah. AT is, pretty standard neurotransmitter. But tyramine, that was the more surprising one, a biogenic amine.
How do they pin them down?
Well, messing with the gene that packages acetylcholine UNC17 suppressed the gut response, so ACCN seems necessary maybe to get the message loaded up.
But tyramine, that was the clincher.
That seems to be the defining component, yes. When they deleted the enzyme that makes tyramine, it's called TDC1, that completely abolished the longevity boost from neuronal XBP1s.
Wow.
And it significantly suppressed the UPRER activation in the intestine, too. So tyramine looks like the critical link. It is the message being sent.
Okay, but that raises a question, doesn't it? Why use a relatively simple molecule like tyramine for such a crucial system-wide response? Why not something more complex like a peptide hormone?
That's a fair point. Peptides, though, they generally take more energy and time to make, transport, regulate. Tyramine is quick. It's simple.
And speed matters in a crisis.
Exactly. The paper suggests this pathway often kicks in response to sudden threats, like running out of food. So speed is probably key. A simple amine is like a fast, decisive, hit the survival mode button, and now W.
Got it. Fast and effective. So tyramine's the driver.
Yeah.
Which leads to the next really cool bit of specificity in the source. Which neurons are actually sending this signal?
Right, because not all neurons make tyramine. The enzyme needed, TDC1, it's only found in a very small, specific set of neurons in the whole worm.
They zeroed in on those.
They did, and the finding is kind of astounding, really.
How specific are we talking? Just a few.
Try 4. The paper showed that turning on XBP1s in only the 2 pairs of neurons that make tyramine, these interneurons called RIM and RAC, that was completely sufficient.
Sufficient for what?
Sufficient to trigger the UPRER activation in the distant intestine and extend the worm's lifespan.
4 cells, running the whole organism's survival strategy.
Yeah.
That's incredible precision.
It really is. The RIM and RAC neurons are basically the command center for this whole response, and XBP1s is the switch flicking them into action.
Okay, but is this just a lab phenomenon, or does the worm actually use this in, real life?
Good question. They check that. Physiologically relevant stress, like a toxin that triggers UPRR, or importantly, starvation.
What happened?
Under those real-world stress conditions, they found that XTP1 was naturally activated, spliced in those exact rimric neurons.
Ah, okay. So the worm's nervous system senses low nutrients, flicks the XBP1 switch in those four specific cells, and bang out goes the tyramine signal.
Yep. The signal basically says, emergency, batten down the hatches, prepare for long-term survival.
And that tyramine signal, it does more than just help with longevity, right? It coordinates a whole suite of changes.
Oh yeah, a whole package of survival responses. Many of them look exactly like what happens during actual starvation, even if there's food around.
Like behavioral changes.
Yeah.
Those are usually pretty obvious signs. What do they see?
Classic starvation behaviors, yeah, driven by tyramine. For instance, worms with active neuronal XPP1s were much, much less likely to leave their food patch, the bacterial lawn.
Risk aversion. Makes sense. If times might be tough, you stick close to the food you have.
Precisely. Conserving energy, reducing the risk of searching for potentially non-existent better options.
And was this linked directly to tyramine?
Absolutely. When they mutated the tyramine synthesis gene, TDC1, that reluctance to leave the food patch, gone. Completely suppressed. Locks tyramine in as the direct signal for that caution.
What about general movement? Do they just slow down?
They did. Less exploration off the food and even a smaller foraging range on the food patch, just less movement overall.
Also conserving energy.
Textbook survival strategy. And again, those changes were reversed if you knocked out tyramine synthesis. Tyramine tells the worm, stop unnecessary wandering, hunker down.
Makes sense. And survival mode often means shifting resources away from reproduction, right? Investing in the future is risky when your own survival is threatened.
You got it. Clear resource management. Activating neuronal XBP1s significantly reduced the total number of offspring, the brood size.
Fewer babies to conserve resources for the parent's own survival.
Likely, yeah. And guess what? That effect was also suppressed when they got rid of tyramine synthesis.
The tyramine controls energy use, risk-taking, and reproduction levels. It's really coordinating things.
But wait, there's more on the reproduction front. It gets even more sophisticated. Neuronal XBP1s also caused a really striking increase in internal hatching, vivipary, late in the reproductive cycle.
Vivipary. The eggs hatching inside the mother.
Yeah.
That sounds dramatic.
It is. It's a protective strategy. The young basically use the mother's body as a temporary safe haven, like a biological bomb shelter.
Giving them a better start in harsh conditions.
The source calls it a potential nutritional springboard. Gives the young a leg up when the environment is really tough.
Wow. Oh, kind of last-ditch insurance policy for the next generation.
And, you guessed it, this dramatic internal hatching, also completely suppressed by losing tyramine synthesis. It just reinforces tyramine's role as this central coordinator for all these complex survival tactics.
Okay, let's recap the pathway then. Stress, like starvation, triggers XBP1s in just those four rimric neurons. They release tyramine. Tyramine is the message.
True.
Now, that message has to land somewhere. Tyramine needs to hit a receptor on the intestine to deliver the instruction and turn on the UPRER. Should be simple, right? Key into lock.
You'd think so. A nice, straightforward activating signal. But this is where the mechanism gets. Well, beautifully complex, actually. It requires some close attention.
Uh-oh.
They focused on a known receptor in the intestine for these kinds of amines called OCTR1.
Okay.
And there are a couple of twists. Twist number one, OCTR1 was mostly thought to be for octopamine, tyramine's chemical cousin, but new data showed it responds to tyramine too.
All right, so the messenger, tyramine, can indeed bind to this intestinal receptor, OCTR1. Makes chemical sense.
Check, check. But now twist number two, this is the big one. Activating XBP1s leads to tyramine release, which then somehow causes the amount of OCTR1 receptor on the intestinal cells to go down.
Wait, hold on. The signal molecule is released, and the result is fewer receptors to receive the signal. That feels backwards for a simple activation cascade.
It's completely counterintuitive, but it gets even stranger. The researchers then showed that if they artificially knocked down OCTR1 levels in the intestine, just reduce the amount of the receptor, that by itself was enough to turn on the UPRR and extend lifespan.
Even without the original neuronal signal.
Even without it.
So, the survival response, the UPR activation, isn't triggered by tyramine activating something new. It's triggered by the absence or reduction of this OCTR1 receptor.
That's exactly it. Think of OCTR1 as being like a brake on the UPR survival pathway.
Okay.
When the worm is happy and well-fed, lots of OCTR1 is present, keeping the UPR pathway quiet, inhibited. Then Stress hits the neurons, XUP1s activates, tyramine gets released.
And tyramine's message to the intestine is,
Cut the brake line. Reduce OCTR1.
the signal doesn't push the gas pedal, it disables the brake.
Precisely. By reducing the OCTR1 receptor levels, the inherent protective mechanism, the UPRER, is finally unleashed. It's disinhibition, not direct activation. Probably involves some baseline inhibitory signaling, maybe through a geocoupled GPCR pathway that gets shut off.
Wow, that's a really elegant, if complex, regulatory switch. Ensures the system only goes into full survival mode when absolutely necessary by removing the constant off signal.
It's a sophisticated solution.
Okay, so that structural shift from thinking activation to understanding disinhibition, that's really the core technical insight here. We've basically uncovered this profound mechanism. Neuronal XBP1s in just 4 cells acts as a stress sensor.
Right. Prompted by things like starvation.
It remodels those neurons to release tyramine. Tyramine then acts globally, coordinating this whole survival response: longer life, better cell maintenance via UPRR, cautious foraging, and that optimized reproductive strategy, including viviprey.
It's a beautiful example of how even simple nervous systems integrate environmental cues into body-wide metabolic control. And the source material draws a really compelling parallel here.
To mammals.
To mammals, yeah. XBP1's activation in certain neurons in our hypothalamus part of the brain is known to link ER stress to controlling metabolism systemically. And guess what those pathways involve?
Let me guess. Something like tyramine.
Very close. Norepinephrine, which is a direct chemical cousin, an analog, to tyramine and octopamine in invertebrates.
So the basic blueprint, a brain stress sensor using a simple amine signal to orchestrate metabolic and survival changes throughout the body. That architecture might be deeply conserved, hardwired into our biology too.
It certainly looks that way. The fundamental logic seems conserved across vast evolutionary distances.
Which brings us to a final provocative thought for you, the listener. Given how effective this pathway is in the worm, using a simple amine signal from a few neurons to coordinate this whole suite of survival adaptations, what might be the potential for tapping into, maybe even manipulating, these conserved neuronal metabolic pathways to promote health and longevity in more complex organisms, maybe even in us.